hadn't been able to verify whether or not our target civilization to have been the Nephites, meaning the Hopewell Mound Building people, had never been able to verify whether or not they in fact have or harbor haplogroup X DNA. In 2008, they sequenced the remains from about 21 individuals who are known to be from Hopewell Mound Builder mounds. When they sequenced that DNA, they found that haplogroup X was in fact detected in 3% of the individuals from a Middle Woodland Hopewell site in Illinois. Remember, Illinois is where Zelf was found. It's also found in 6% of the Norris Farms individuals. This is low percentages, 3 and 6%, but this is also an incredibly small sampling. Only 21 individuals have been sampled. The fact that it is there at all is what is really significant to me, is that we have now the target civilization to have been the Nephites do in fact have DNA that is found also in Jewish populations and in the Druze population over in the Mediterranean, which again then goes in support of the truthfulness of the Book of Mormon, although it does not at this point in time prove it. So that we have to be careful again not to overstate what we have found here, but it certainly is looking very good for the claims of the Book of Mormon from a genetic standpoint. Now, we need to address the primary challenge with haplogroup X being related to the Book of Mormon, which is its date. When did it arrive in the Americas? According to American Journal of Human Genetics 2003 article, it says, when the sequence variation of the first hypervariable segment of the control region is analyzed, haplogroup X mitochondrial DNAs from Europe and the Near East are found to yield similar coalescence times, 17,000 to 30,000 years before present, and 13,700 to 26,600 years before present, respectively. That's the haplogroup X DNA coming out of Europe. By my understanding of the scriptures, and specifically Doctrine and Covenants, section 77, if haplogroup X arrived in the Americas 30,000 years ago, that is about 26,000 years before Adam was on the earth, which I find very difficult to understand how that could be. What a lot of people don't seem to know, especially in the LDS scholarly community and other groups who are taking issue with haplogroup X being related to the Book of Mormon, is they say, well, haplogroup X was here long before Lehi and his group because they showed up here 30,000 years ago, but seem to be unaware of the fact that there is a huge debate going on in genetics right now about this very dating issue. So we need to take a very close look at the dating. This is from Annual Review of Genomics and Human Genetics 2005 article. They showed that a model that includes a rate variation in the mitochondrial DNA control region gave an estimate for the age of the human mitochondrial DNA ancestor that was half of that obtained when a single mutational rate was assumed. When you get a date or a rate that is off by half an age, that really is not a very strong or robust dating. Therefore, average age estimates of the mutation rate for human mitochondrial DNA do not reflect the true state of affairs and should be viewed as simplistic tools for phylogenetic studies. Now remember, we have phylogenetic dating and then we have pedigree dating, which we already talked about. But then he goes on to say this. This means that care must be taken when using mitochondrial DNA data to date phylogenetic events, as the underlying assumption of neutral clock-like evolution may not hold. It's not like every 6,764 years, there all of a sudden, bleep, there's a mutation. And then 6,700 more years, there's another mutation. That's kind of the assumption that happens when you're extrapolating from 6 million years of the human-chimp species split. This is 2005 article in Molecular Biology and Evolution. This is called The Time Dependency of Molecular Rate Estimates and Systematic Overestimation of Recent Divergent Times. The most marked differences have been seen between rate estimates directly measured from pedigree or population studies and those that are inferred in phylogenetic species-level studies. The large rate disparities present a direct challenge to the neutral hypothesis of molecular evolution. The theory that is being postulated based on phylogenetic rates isn't matching up in other species, not just the human species, but other species as well, and doesn't match up with the pedigree rates when they actually take reality dating from one organism to the others. So this is actually creating a direct challenge to the claims of the neutral hypothesis of molecular evolution. American Journal of Human Genetics, 2004 article. By quoting the number of mutations in the branches of a haplotype network from samples of Native American populations. Now this is now bringing it back to the Native American populations. This article states that they found a striking difference between their evolutionary estimate of 2.6 times 10 to the fourth per 20 years and the pedigree estimate. Again, saying that there's a big difference between what would be considered their evolutionary estimate and from what they're finding in reality. They go on to say that it is unclear which rate should be used for evolutionary studies. We need to know those mutations that are involved in differences between lineages or populations. An inappropriate choice of the mutation rate value may produce a tenfold deviation from the true age of past population events. This discordance between the two kinds of estimates needs to be addressed. This is in 2004. They're saying there's a huge discrepancy between what we see from the reality dating and what we are getting from the theoretical dating. This is from Systematic Biology, 2007. This gives another example of what I'm talking about here. Howell in 2003 obtained a pedigree divergence rate of 1 mutation per base pair per million years for the control region. Building on these data, Howell also combined data from a number of other unrelated pedigree studies and obtained a broadly similar control region pedigree divergent rate of 0.95, which is close to 1, of course. Then Ho points out that this value is vastly greater, approximately 50 times greater than the phylogenetically derived divergent rate of approximately 0.02 mutations for protein-coding mitochondrial DNA. So again, the phylogenetic theoretical-based dating is 50 times different than what they are finding in the reality dating. Here's in the journal Heredity, June of 2008. In the News and Commentary, this is a very descriptive title. It says, The Molecular Clock Debate. Now this is June of 2008, and this debate is raging right now. Time dependency of molecular rate estimates for mitochondrial DNA. This is not the time for wishful thinking. It says, firstly, there is a steady accumulation of reports that human mitochondrial DNA does not evolve in a clock-like manner. Sequence sets that pass a robust clock test are the exception rather than the rule. Secondly, we lack adequate calibration points for accurate time estimates, even if there was a human mitochondrial DNA clock. There was an individual by the name of Dr. Bandelt who essentially called the pedigree dating geneticists on the carpet and said, you guys are messing things up. You're not doing something right because our theoretical phylogenetic-based rate is different than what you keep getting. And so he kind of fired a shot, if you will, over the pedigree dating people. And the pedigree dating people are writing this article back. Dr. Bandelt, again, takes the opportunity to dismiss the discrepancy between mitochondrial DNA rate estimates from pedigree analysis and those from phylogenetic analysis. We disagree that the pedigree rate is not well defined, which, of course, Dr. Bandelt had asserted. It has an explicit operational definition and is, in fact, more empirical and less model or theory dependent than phylogenetic rate estimates. So they're saying, wait a minute, our rate estimates are not mistaken. It's your theory that's messed up. Okay? So then they go on. It says, Dr. Bandel also makes the unsubstantiated charge that pedigree analysis seems to suffer from ascertainment bias and sequence errors. We can't find evidence for either of these issues he raises, and they've been addressed previously anyway. Our meta-analysis confirmed that the pedigree rate was less than one set of phylogenetic rates by an order of magnitude. A more significant problem is that the phylogenetic rate estimates vary widely, which is something that should trouble stopwatchers. Now, they're calling stopwatchers people who are saying that